Hello and welcome to our chapter on DNA replication. The important points that you'll want to understand or remember from this chapter are listed here. We have the DNA structure, so just basic features of DNA structure we've already really discussed in a previous chapter, but they're really going to be important for you to understand as we go through the replication process. And then we have the main highlight of this chapter, or the main focus of this chapter, which would be the process of replication itself. So understanding how that process works and what proteins and enzymes are involved in building these new strands of DNA. Also some key terms here, the replication bubbles and fork, and then the difference between a leading and a lagging strand is very important to understand. And then DNA repair mechanisms, also very important to understand the proteins and enzymes involved there. And then finally, understanding how DNA is packaged in the nucleus in order to fit. There are levels of packaging that the DNA undergoes. All right. So first, we'll go on to the structure. So this is a review, really, from back in our chapter on macromolecules. And we have our two DNA strands shown here with the sugar phosphate backbone, and then our nitrogenous bases pairing up in the center of the molecule with hydrogen bonding. And we notice we have our numbering here on our sugar. Remember, we start numbering the carbons on the sugar where the sugar meets the nitrogenous base, so that would be carbon 1, 2, 3, 4, and 5, and all the other ones would be numbered in a similar way. So when we look at the molecule, this is the 5' end of the molecule because the 5' is toward the very top, or the uppermost part of the molecule. And if we look down here, if we were to number this carbon, or these sugars, the carbons in this sugar, we would say 1, 2, 3, 4, 5. So this is the 3' end of the molecule. Okay, and this is the 5' end. Now, the DNA molecule is such that the other strand, the opposite strands, runs in the opposite direction, or has opposite strandedness. So here we see, if we look at this strand on the other side that's pairing up with, we have carbon 1, 2, 3, 4, 5. You always number in a clockwise direction. And we see that this is the 3' end, because it's toward the top. We're always looking at 3 versus 5, only 3 versus 5, because that's where new nucleotides, or the attachment points between nucleotides occur as far as bonding. So this is a 3' end, and if we look down here, we'd number this one, 1, 2, 3, 4, 5. This would be the 5' end. So they run in something called an anti-parallel direction. They're parallel, but they're running in opposite directions. Also, something I want you to notice here is that on the 3' carbon, remember 1, 2, 3, we have a hydroxyl group. Okay. On the 5' carbon, we do not have a hydroxyl group. We need that hydroxyl group in order to add another nucleotide. That hydroxyl group is very important. It is of the utmost importance to catalyze that reaction of adding a new nucleotide. All right, so that's the basic overview of this structure. Let's talk more about this replication process in general. So here we have our DNA molecule, just a simple kind of ladder-like arrangement. And we have our nitrogenous bases pairing up in the center. And what will happen is an enzyme, which we'll talk about, will pull the strands apart, because remember, this is just hydrogen bonding. It's very weak. We'll have two templates to work with. Our new nucleotides will be brought in by another enzyme, and they're shown here in light blue or turquoise. And they're brought in to complement whatever nucleotide or nitrogenous base is shown on the strand. And then we end up with two daughter or two new DNA molecules as a result. And each has an old strand and a new strand. Okay. So some more key terms here with replication. We have our replication forks and replication bubbles. So here we have our old strands in dark blue, the new strands forming in light blue, and we see along the DNA molecule we have several points of origin of replication, or replication occurring at several points along the molecule, and this is to allow the process to occur in a timely fashion. If you just had one replication bubble and you had the enzymes working in just that one area and kind of moving to either side, it would take a lot longer than if you had several teams of enzymes working on this DNA molecule at a time. So we have several replication bubbles, and at the end of each bubble we have a fork. So here's one fork, here's another fork, and the fork really just represents the direction of the movement, or where the enzymes are really ultimately moving toward adding these new nucleotides. So they add them on really both sides of the fork. I'm sorry, both sides of the bubble. Now we're going to be focusing in on this side of the replication bubble, this particular replication fork on the left. And this replication fork is shown here, just the fork. Of course it would extend out to the bubble, but we're really going to focus here on just this fork, and to understand the overall process of replication. So looking at these two strands, we have our old strand, and you see here in dark blue, and then the new nucleotides being brought in the turquoise, with the light blue for our new strands. And we have something called a leading and a lagging strand. The leading strand is called the leading strand because it leads, or it's produced faster or replicated faster than the lagging strand. The lagging strand synthesis lags behind the leading strand, and we'll see why that is as we go through the process, and it'll become very clear. But just to take a look at this process, leading versus lagging, here's time A, here's time B a little bit later. We can see the leading strand. We have our enzyme that's adding these new nucleotides. This kind of pinkish thing is the enzyme adding the turquoise nucleotides here. And you can see the lagging strand, it's a little slower, and then time B, leading strand is just cranking along, adding those nucleotides, whereas down here we're seeing it's kind of moving along, and we're moving toward that fork, but it's taking a little bit longer, and it has to do with the nature of the DNA molecule and the strandedness that's involved. So the key enzymes and proteins involved in this replication process are listed here in the relative order that they come into play in the process. And it's important for you to understand not only the order that they act in DNA replication, but also their purpose. So we have our helicase, and that is going to unwind the helix and separate the nitrogenous bases so that we can bring in these new nucleotides. And SSB, it stands for single-stranded binding proteins, and these prevent the strands from coming back together because they are really held together by hydrogen bonds, so their affinity for one another is great. So these proteins prevent them from coming back together during the replication process. And then we have something called primase, which adds a short sequence of actually RNA, a tiny short sequence of RNA to the DNA template, so that our next enzyme, DNA polymerase III, can add nucleotides to the new strand, to this little tiny piece of RNA to kind of prime the new strand. And we'll talk about the reason for the need for this primase as we work through the process of replication. And then the next enzyme to come into play is our DNA polymerase I, and this is an enzyme that is going to replace the RNA that was laid down by the primase with DNA nucleotides, because we don't want RNA in our DNA. So it kind of cleans up that part. And then finally we have something called ligase, and this is an enzyme that is going to seal together the fragments in the new strands, the fragments of DNA in the new strands. And that'll be very useful, or used a lot more in the lagging strand than in the leading strand, but it is used to some extent in the leading strand, and again more so in the lagging strand. So we'll stop this video here and we'll continue on with the replication process in the next video.